Hi everyone, we are here today to show you the wonders of fluid balance. Life is all about balance. There is good and evil, dark and light, and filtration and absorption. Peace, or good grade, can only be found when balance is achieved.
Fluid balance is the relationship between filtration and reabsorption in the capillaries and tissues. Our subject is a frog with an open circulatory system.
What do you mean by an open circulatory system, boy?
An open circulatory system is when blood in an organism's body is not contained within arteries or veins, and there may even be an opening to the outside environment. In this case, the ventricle was open so the fluid could leave the system.
Whoa, that is all very interesting, but you're going to have to slow down. Arteries and veins? What are those?
An artery, similar to your heart, pumps oxygenated blood throughout your body. They are composed of elastic elements and smooth muscle that allow them to contract and expand according to the body's needs. Veins carry deoxygenated blood back to the heart. They also are composed of smooth muscle to help them contract and expand.
That surely can't be all of the components of a circulatory system, right?
Correct. There are also things called arterioles, capillaries, and venules.
English, please.
Sorry. Arterioles are the extensions of arteries that lead to capillaries. Capillaries are between arterioles and venules. Venules transport the blood from the capillaries to the veins so that it can return to the heart for oxygen.
How do all of these relate to fluid balance?
Arterioles and venules can contract or dilate, changing the radius to affect the rate of blood flow through them, into and out of the capillaries. Capillaries can filter some parts of the blood into the body's tissues. The result is a decrease in fluid volume in the circulatory system. Alternatively, capillaries can reabsorb fluid from the tissues, resulting in an inverse effect.
I think I am finally starting to understand. There wouldn't happen to be any equations that explain the relationship between filtration and absorption, right?
Funny enough, there actually is. Starling's equation of ultrafiltration explains this relationship. It states that the rate fluid is filtered across vascular endothelium is due to the amount of pressure between the capillary and interstitial tissues. Below is the equation in its written form.
Very interesting. There are some symbols that I have never seen before. Can you please explain those?
Yes, let me explain. The first symbol is the flux of water across the capillary's walls. K is the filtration coefficient. Pc is the capillary hydrostatic pressure. Pi is the interstitial hydrostatic pressure. Pi is interstitial oncotic pressure. Pi c is the capillary oncotic pressure.
What do you mean by filtration coefficient?
The filtration coefficient is a measure of the membrane's permeability to water. In other words, it measures the volume of fluid that is filtered over time through a known amount of area on the membrane under differing pressures.
I see. The picture is becoming clearer now. Can you tell me what oncotic pressure and hydrostatic pressure means too, please?
But of course, lad, oncotic pressure, or colloid osmotic pressure, is a type of osmotic pressure generated by proteins such as albumin and other large non-penetrating solutes. A non-penetrating solute is a molecule that is too large to pass through the membrane. Hydrostatic pressure is the pressure that is generated from water.
Very cool. I feel like a smart frog now that I know Starling's equation of ultrafiltration and the meaning of every component. Can you give examples of what happens when the equation's variables change?
Sure thing. An increase in capillary hydrostatic pressure or interstitial oncotic pressure results in an increase in flux across the capillary wall. The inverse also holds true. Alternatively, an increase in interstitial hydrostatic pressure or capillary oncotic pressure results in a decrease in flux across the capillary wall. The inverse, again, holds true.
Neato! I think that makes perfect sense. I understand now how the balance between filtration and absorption can be a fine line. Are there any other equations to explain other factors that affect this balance?
Yes, there are two more equations that help to explain more of this process. Ohm's law and Poiseuille's equation are used to describe mechanisms of bodily fluids. Ohm's law demonstrates how the rate of flow of a fluid is derived from the change in pressure applied in relation to the resistance present. It is written as Q, or flow, equals change in pressure over resistance.
And how does Poiseuille's equation come into play with Ohm's law?
Poiseuille's equation states that the resistance equals 8 times the length of the blood vessel multiplied by the viscosity of the blood, divided by pi multiplied by the radius of the blood vessel raised to the 4th power. I understand that can sound quite confusing, but I assure you it is not.
Okay, so there is Starling's equation of ultrafiltration, Ohm's law, and Poiseuille's equation that are all used to explain fluid balance?
That is correct. The three of these equations are very useful in determining mechanisms behind why things change during experimentation. For example, in many fluid balance experiments, the drift rate and weight change are measured. These equations are very useful in explaining how and why these two measurements vary under different circumstances.
What type of circumstances do you mean, Dat?
For our experiment, we tested 3 different chemical compounds to see their effects of fluid balance. The The chemical compounds were adenosine, histamine, and acetylcholine.
Adenosine, histamine, and acetylcholine? This sounds like some sort of foreign boy band or something.
Haha, no Pepe. These are chemicals that have specific effects on the body. Adenosine is a potent vasodilator that can be used to treat types of supraventricular tachycardia. It is a purine nucleoside that is made up of adenine attached to a ribose sugar molecule. It is commonly found in nature in forms such as ATP, ADP, or CAMP. Adenosine triphosphate, adenosine diphosphate, and cyclic adenosine monophosphate are all used in biochemical processes throughout our bodies.
In order for adenosine to act in the body, it must be received by some sort of receptor, right?
Yes, Pepe, that is correct. You are a very smart frog. There are 3 receptors that adenosine can bind to on smooth muscle arterioles. A1, A2, and A3 are the receptor subtypes. Each receptor is a G protein coupled receptor. The A1 and A3 receptors are coupled with the GI pathway and cause vasoconstriction. The A2 receptors are coupled with GS pathway that causes vasodilation, which was what we found in the experiment. The GS pathway starts with the G protein receptor A2 being activated from the adenosine ligand. The G protein alpha subunit is activated by the A2 receptor and it stimulates adenylate cyclase activity. An increase in adenylate cyclase activity increases CAMP levels and in turn also increases pKa levels. The pKa level increase causes myosin light chain phosphatase, or MLCP, to dephosphorylate myosin. When myosin phosphatase dephosphorylates myosin light chains, it causes a decrease in myosin ATPase activity. A decrease in myosin ATPase results in less hydrolysis of ATP and less muscle tension. Additionally, the pKa level increase activates K+ channels on the membrane.
Is histamine similar at all to adenosine?
It is similar in that it also acts as a vasodilator and has 4 receptors that it can bind to. However, the chemical is an organic nitrogenous compound that is derived from the decarboxylation of the amino acid histidine. The 4 receptor subtypes are H1, H2, H3, and H4. Our experiment suggests that the H2 receptors were targeted and it went through a GS pathway to vasodilate. The pathway is the same as adenosine's. If the weight and drip rate decreased, we could say vasoconstriction of the smooth muscles of the arterioles occurred. This would target the H1 receptors that go through a GQ pathway.
I see now that adenosine and histamine can both be vasodilators. Is acetylcholine the same?
Acetylcholine is a neurotransmitter that is released from the parasympathetic autonomic nervous system. It acts on muscarinic 3 acetylcholine receptors, or M3-AChR, in smooth muscle. The receptor follows the GQ pathway and results in contraction from the excitatory effects. The GQ pathway begins with the acetylcholine ligand binding to the M3-AChR receptor, activating the G protein. The G protein activates the amplifying enzyme phospholipase C, or PLC, and PLC activates inositol triphosphate 3, or IP3, in the cytoplasm. IP3 activates ryanodine receptors on the endoplasmic reticulum, allowing for calcium to be released into the cytoplasm. Calcium can then bind to a calmodulin, activating myosin light chain kinase, or MLCK. MLCK is responsible for phosphorylating myosin light chains in myosin heads. As a result, myosin ATPase activity increases, causing myosin cross-bridge formation and an increase in muscle tension. PLC also changes phospholipids in the membrane into diacylglycerol, or DAG. Protein kinase C, or PKC, phosphorylates proteins once it is activated by DAG. PKC also activates long-type calcium channels, or LTCCs, to increase the amount of calcium in the cell. In other words, acetylcholine can cause vasoconstriction of smooth muscle and arterioles.
Okay, so how does adenosine, histamine, and acetylcholine affect drip rate and weight change? I bet we were going to use some of those equations from earlier.
You are correct. The relationship between Postay's equation and Ohm's law is seen here. The result of vasodilation is an increase in the radius of the blood vessel. According to Postay's equation, when the radius increases, the resistance decreases. According to Ohm's law, when the resistance decreases, the flow rate increases. The weight increases due to Starling's law favoring filtration, in which the flux is positive. The inverse holds true in regards to vasoconstriction as well.
Very neat. I am thoroughly enjoying learning about all of this. I find it interesting how fluid balance can be greatly affected by vasodilation and vasoconstriction. More specifically, what effect did the chemical compounds have during experimentation?
Well, before I tell you about their effects, let me describe how we begin the experiment. Before each chemical treatment, we ran a control using Ringer solution with 3% dextrin. The Ringer solution replicates the general composition of ions in bodily fluid. The dextrin replicates the absence of albumin, a protein that increases oncotic pressure within capillaries because it is a non-penetrating solute. The control helped to establish a known drip rate and weight so that we could determine the exact effects of each treatment.
You could say balance is all about control.
Haha, that is very true, Pepe. You are so wise. After perfusing with the control and establishing a known weight and drip rate, the treatment was performed. Adenosine and histamine resulted in an increase in drip rate and an increase in weight. Acetylcholine resulted in a decrease in drip rate and decrease in weight.
So if I'm understanding this all correctly, Adenosine and histamine caused the vasodilation of arterioles, causing the resistance to decrease while flow and filtration increase. And alternatively, acetylcholine caused the vasoconstriction of arterioles, resulting in increased resistance and decreased flow and filtration.
You make me proud, Pepe. I feel like I have taught you well. That is correct, and you are very smart, Froggy. Take with you this knowledge and bring balance to the force, your life.